Good morning, everyone, and welcome back to another session of our A&P. We're going to do today is we're just going to finish up Chapter 12. Your exam, which will be next week, will cover Chapters 10 and Chapter 12 in their entirety. As I posted on Blackboard, your quiz is going to be an online quiz. For this particular time, as I told you all last week, my children were all sick, and they like to cuddle when they're sick, and so when they cuddle, it kind of makes the people they cuddle with sick. So now I've got whatever they had at the strep throat, and I don't think I have an ear infection yet, but I'm pretty sure I have strep throat, and I'll get a confirmation of that later on today. Anywho, so for my Monday classes, your labs, I've called the biology department, and they should be open for you at both 3 o'clock and 5 o'clock so that you can go in there and study. For my Tuesday, Thursday classes, you will still have your quiz, and it will still be online, and I will send an email posting it to all of you guys when the quiz is live and when you're able to take that quiz. And from the time that I make the quiz live, from the time you receive that email, you'll have about 24 hours to take the quiz. It will be timed. You'll have 45 minutes. And I've only timed it to make sure that you guys aren't looking up all those answers, because I have a feeling that you're probably trying to look up some of the answers, but I don't want you looking up all of them. So it will be timed as a result. Next week, we will have our exam. My hybrid classes, it'll be sort of the same thing. If there are times outside of, the time that I have allotted that you need to take your exam, let me know. All right, so finishing up with information processing, remember that the neurons send and receive those messages, and how that neuron responds to that message, and the messages are sent via chemicals called neurotransmitters and neuromodulators, and how those neurons respond to them is going to be based on the type of receptors that are there. So when we talk about the simplest level of information process, we're just talking about individual neurons themselves. So the receiving end of the neurons, the dendrites, are going to receive many different neurotransmitters at the same time. Some of those neurotransmitters that they receive are going to be excitatory, which means they will cause action potentials. Others will be inhibitory. So the net effects on how that neuron receives that message is going to be what determines if an action potential is received. If we have more inhibitory neurotransmitters that are received on the receptors than excitatory neurotransmitters, then an action potential will not be produced. Postsynaptic potentials are graded potentials that develop in the postsynaptic cell, the receiving cell, and they have these graded potentials in response to neurotransmitters. And that's not really anything all new. In fact, the last few slides that we go over here, they're all just to kind of tie everything together on what we've talked about as far as the receiving cell and the sending cell in neural processing. So we have two types of what we call postsynaptic potentials. And if you just look at the term postsynaptic potentials, post means after, synaptic, so the synapse. So the postsynaptic cell is always the cell that's receiving the message. And then the potentials on there, you kind of start thinking of action potential and resting potential. So the two different types of postsynaptic potentials will be those that will give you an action potential, which we would call excitatory postsynaptic potentials. This is where that graded depolarization of the postsynaptic membrane will lead to an action potential. The neurotransmitters that are released are going to bind to more receptors that will allow sodium ions to come in to trigger the action potential than any of those inhibitory neurotransmitters that bind to those receptors. Conversely, if we have inhibitory postsynaptic potentials, then we're going to have hyperpolarization of the membrane. And every time you see postsynaptic membrane here, I want you to think the receiving end of the cell. So an inhibitory postsynaptic potentials, the receiving end of the cell is going to have more of these neurotransmitters or chemical messengers that say, hey, don't generate an action potential. In fact, I want you to get as far away from an action potential as possible. And we know that the more negative you go, the further away you get from generating an action potential. We also understand that it's the movement of potassium ions outside of the cell that gets us even more negative. So if we have lots of inhibitory postsynaptic potentials or neurotransmitters that are telling the cell, I don't want you to generate an action potential, I don't want you to do that, what will happen is that the neurotransmitters will bind to the receptors on the receiving cell or the postsynaptic cell, and it will open potassium channels. And those potassium channels opening up will kick out a lot of potassium. So in kicking out all of that potassium, we now become hyperpolarized or get down to negative 90 millivolts, which makes it more difficult for an action potential to take place. So if a neuron receives many inhibitory postsynaptic potentials, that's what all that stands for, then that neuron is going to be inhibited from producing an action potential. And that's exactly what we said before, because the stimulation needed to reach threshold is increased. All that really means is that we've gotten so far negative, it's going to take us a lot of stimulation. It's going to take a lot of sodium ions. It's going to take a big stimulus to get us back to negative 60, negative 55, or what we consider threshold. In summation, we want to trigger an action potential, and typically one excitatory synaptic potential isn't going to be enough. So a lot of times, and we've just kind of been talking about this very generally, that neurons release neurotransmitters and they bind to the receptors of the postsynaptic cells, and it tells the postsynaptic cell to do something. You have to keep in mind that very rarely is it one neuron to another neuron. Typically, there are several neurons that are communicating with one neuron at the same time. So there are lots of messages that are being sent and received to one neuron at any point in time. And how that neuron decides, and that's really what all this is saying, this last little bit of the notes, how that neuron decides whether or not to generate an action potential or to not generate an action potential is depending on the accumulation of which of these excitatory or inhibitory potentials are in abundance. So the more excitatory potentials you have, the more likely you are to generate an action potential. If you have lots of neurons that are telling you don't generate an action potential, they're releasing GABA or other neurotransmitters that that don't allow that to happen, that are inhibitory, then we won't have an action potential. So a neuron becomes facilitated when the excitatory postsynaptic potentials accumulate. So that will rise the transmembrane potential closer to threshold, which means more sodium ions come in, and this happens until a small stimulus can trigger an action potential. So it might just be just a little bit of an excitatory neurotransmitter that's released and received by this receiving cell that allows for an action potential to take place. But keep in mind that there are lots and lots of these excitatory postsynaptic potentials that have taken place, that have been received by this receiving cell, the postsynaptic cell, that will allow for this to happen. Neuromodulators and hormones can change membrane sensitivity to neurotransmitters. So if there are neuromodulators, such as opioids, that are released from one cell and are received by the other cell, the postsynaptic cell, then it can make the membrane more or less sensitive to other neurotransmitters. And when that happens, that's going to change the balance between excitatory and inhibitory postsynaptic potentials. So it's a lot more complicated. We're kind of just scratching the surface of synaptic activity. Many scientists and researchers to this day say the brain and the nervous system is the final frontier of the human body. It's one of the only organ systems that is still pretty poorly understood. We don't know as much about it as we possibly can. We just understand that it's a very complex process. So if this last little bit of the notes don't really make as much sense to you, they're not really painting as clear a picture as maybe some of the other parts of the notes did, there's a reason for that, because we're still not quite sure exactly how all of this is going on. We just know that there's this interplay of hormones and chemical messengers we call neurotransmitters and neuromodulators that allow for these cells to communicate with one another and to process information to give us the appropriate results or the appropriate response to the information that's being sent and received. So this last little slide here is just a picture that shows that very rarely do you have just one cell talking to another cell. As you can see here, there are 3 cells, and this is still quite an oversimplification of this, where at any point in time there can be dozens of cells that are communicating with one another or with another cell. So we have here our yellow cell that the action potential arrives, and all of this information you're very familiar with. Calcium comes in, and the amount of calcium that comes in is going to trigger the release of a neurotransmitter. But from this green cell that's talking to this yellow cell, an inhibitory postsynaptic potential or an inhibitory neurotransmitter, GABA, has been released, and notice it has an effect on the calcium channels. So as GABA is released, it activates those calcium channels, and less calcium is going to come into the cell, which means that less neurotransmitter is going to be released, from the cell. And if you have less neurotransmitter that's released, then it's going to reduce the effect of, it's going to create an inhibitory postsynaptic potential on this postsynaptic cell, this purple cell that's receiving. Conversely, if we have serotonin that's released, which is going to increase the amount of calcium channels that open up, more calcium enters the cell, more neurotransmitters are released, so this purple cell has been facilitated, or it has an abundance of excitatory postsynaptic potential, so it will generate an action potential, whereas this cell will not enough neurotransmitters released, and this cell will have plenty of these neurotransmitters that are released, and they will allow more sodium to come into the cell to generate an action potential in the purple cell. All right, that is all I have for today. I probably won't get your quiz posted until later on this evening, because I feel like I am going to die. Ugh, so horrible. So I will get all of your things posted, your quiz posted definitely by this evening, but it might be 5 or 6 o'clock. So just be looking out for the emails during that time for your quiz. And remember, you have 24 hours to take it, but once you log into it, you will not be able to log out of it. You'll have to take it in one sitting, and it will be timed. All right. And I will hope to see you all very soon and not deathly ill. Have a great day.